Thank you for having me. This is my first time presenting at this event. We just came across this event a few months ago, and we figured it'd be a good idea, as we're a Swiss company. We're about to launch our product in the United States. We're 510K enabled, cleared, I should say. We don't do much in the UK yet, so this was a great opportunity to introduce you to some of the people on my team. We have some local boots on the ground, and the UK is a complete virgin territory for us, and we're very much looking forward to learning more about what it is we do over here and how we could actually implement PSP across your various trusts in the UK. So basically, I'm going to talk about enhancing sepsis management. Sepsis management is a broad term that means everything and nothing. But basically, I'm just going to introduce the use of a new biomarker. It is one tool among many tools, and it doesn't mean to be the tool that will solve everything, but I think it's an important part of the architecture of any sepsis management strategy that you may or may not have at this point. Basically, some quick numbers. You know these numbers better than me. The numbers are staggering in terms of the sheer cost of sepsis, but I was most impressed by the societal costs associated with sepsis, and people don't think so much about this. We think about the deaths, the prevalence of cases, the incidence of cases, et cetera, but at the end of the day, these numbers are staggering, and if I can move over to the US for a second, the numbers are even bigger. The numbers are even bigger because all in, if you consider what's happening around the world, 50 million cases is huge. There's only 2.5 million cases in the US. And there's roughly, it's a little under 3 million for Europe as a whole. It's always hard for me to figure out if it's EU-wide. Does that now include the UK or not in those numbers? It probably doesn't. At the end of the day, we have more sepsis cases in Europe, but I believe that we're simply undercounting sepsis cases in the US. And at some point, from a per capita basis, we're more or less aligned. So I tend to do my business planning in terms of what I see in the US, but I'm actually applying it to some European numbers, which I think are closer to the reality of sepsis cases in Europe. Hospital costs 52 billion. That's a big number. So if I go back to the UK number, sepsis costs the UK 2 billion. In the US, it's a 52 billion issue for hospitals alone. Societal cost is actually $62 billion. And what's interesting, if you consider where sepsis cases take place, we understand it could be infants and it can be the elderly. And at the end of the day, reimbursements in the States is very important, as it is in the UK, very different system. But 70% of reimbursements are covered by what we call Medicare and Medicaid, which is effectively insurance for the poor and for the old. So the US government has recognized that sepsis on a national level is a major issue to be dealt with. Cost per case, $35,000. That's how they've come up with some of these numbers. Important in the US, I'm not sure what the numbers would look like in the UK, but readmission costs, roughly $3.5 billion. When you miss an infection at patient presentation and you send these people home and they come back, the insurance companies basically deduct that from your insurance. So, the insurance companies in the US, it's a very specific system, will actually penalize you for having missed an infection when you had a patient in front of you. And missed infections is actually the third reason for malpractice suits in the States. So again, I'm just giving you this as background information for how we think about this. Very much focused on the UK now. We're doing a lot of work in Europe. We're Swiss-based, but I'm also very much involved in all of our development in the States. Voila, okay. Sepsis is a time-sensitive emergency. You all know this probably better than I do. What's interesting is we did a lot of work in the ICU, but it turns out that most patients, most sepsis cases end up walking through the ED, or the A&E as you call it. 85% of sepsis cases present in the ED, yet they're not necessarily screened for. And I think this is where part of the issue is. I think this needs to become a screening tool more than anything else. And we all know that every hour of delayed treatment means, reducing the odds of survival by 8%. You know the statistics. A lot of these statistics do come from the US, and I realize we use them over and over. But I think we'll all agree on what they mean. Basically, our company vision, if I could spend a second on this, is we believe that nanofluidics can transform diagnostics. And nanofluidics is a particular use of technology. It means that we use nanofluidics, basically, nano quantities of fluids to do rapid testing, rapid detection, rapid testing with the use of our Abioscope platform. For those of you who are remotely interested in engineering, this is what a capsule looks like. And effectively what happens is, because we've reduced the distance using the nanofluidic platform, it doesn't, we've got something like a 2-minute incubation time for the actual test. So it's a 7-minute test, of which 2 minutes is part of the incubation process. This is an ELISA in a box concept. And if you've all used ELISA tests in the past, you'll realize that the technology has vastly improved, and applying this using nanofluidics makes it even more performing. Pancreatic stone protein. I'll spend a few minutes on what that actually is. But what we effectively do is, we run it exclusively on the Abioscope. We have an exclusive global license for the pancreatic stone protein, and it only runs at the point of care on an Abioscope. Using 50 microliters of blood, takes 7 minutes, and you get a result. PSP, for those of you who don't know what that is, we've got over 100 plus publications. We've got 2 large multi-centric studies that were performed in Europe and in the US, and we were FDA cleared in November of last year. Was CE marked, of course, IVDR, and of course, ISO as well. I never know where I'm pointing at. Okay, so what exactly is pancreatic stone protein? It's an early warning marker for sepsis, secreted by the pancreas. There's an organ dysfunction that begins caused by some infection. We don't know what the infection is at this point. There's some form of remote sensing, and the PSP is released both from the pancreas and the intestines into the bloodstream. And that's what we effectively measure. So there's a lot of studies right now to try to help us understand, why does the pancreas react? That's not what I'm going to debate today. We are here only to show you how, by measuring PSP levels in the blood, we can actually start to address the sepsis issue. So the PSP cutoffs of this particular marker from our FDA instructions for use are basically anything under 100 nanograms per milliliter is a low risk of sepsis. We're trying to keep it very simple. And then you've got that intermediate space, orange, between 200 and 300. Anything over 300 is a very high risk of sepsis. And this is what we're trying to promote here. Anybody that gets tested, it's about detecting it quickly and activating those bundles. That's giving you a bit of a hint on where we're going with sort of our marketing communication. And I'm going to show you PSP now in reference to some classic usual suspects, as I call them: PCT, CRP, and PSP. Basically, sorry, I keep pointing the wrong way. Okay, so this is an example of a study that was run on 14 sites. We had 243 ICU patients, and we have 53 sepsis cases. This chart on PCT shows some elevated points 3 days before recognition. The blue part is non-septic. There's a lot of overlaps. It's actually not that conclusive. If I go to the next one, CRP, I'd say it's even worse, because there's almost so much overlap. It's very hard to tell whether or not there's an infection. Is it septic? Is it not septic? And the numbers aren't that telling, although there is a rise in the amount of CRP in the blood 1 to 2 days prior to an actual onset. With PSP, the numbers are a lot clearer. We've got a clear blue line, that's non-septic. That shows us basically that effectively it's a very flat line. So if you have a low volume of PSP in the blood, the predictive value is negative. there's a high probability that this patient will not get sepsis. So we're very confident about that based on such numbers. On the other hand, however, and this is where you see this clear discrimination, where PSP levels start to rise, that's a clear indication that there's an onset of sepsis. And we're able to find this out up to almost 3 days prior to clinical recognition. And this is a very, very powerful statement. That effectively means that, we can test someone coming in, and we're able to detect 3 days earlier before a clinician would actually detect sepsis and recognize it and order a bundle that something needs to take place. We're not suggesting necessarily what needs to take place, but we're just suggesting these are patients that you need to take a close look at. This was done on a burn study where there was a clear discrimination between non-infection, the black line, some infection, and then sepsis and septic shock. Clearly, the yellow and the red line are clear indicators. In this case, we're making big push in burn ICUs. We're trying to enter the market in Europe and the UK as well on the burn ICU front, simply because it's going to help physicians actually distinguish between basic infections and actual septic infections. I think that's something that's important to point out at this point. All right, so now this is my big story. Can PSP effectively screen for sepsis in emergency care? Because we believe this is where we need to actually apply this. If 85% of septic cases walk through the front door of a hospital, that's where we need to screen for it. Patient presentation, this may differ from the way you do things in the UK. This is based on a US model, but I think overall it's very similar. Basically, triage nurses don't really know who's walking in. You're wondering what's wrong with this patient. You're not sure. There's some signs, but you're not sure. Basically, you run a few scores like QSOFA and USE2 in the UK, I believe is a more prevalent risk assessment score. And then you're effectively using an ESI system in the States. You stratify your patients. In the case of QSOFA, we ran a study using QSOFA, which is I'm using it here. Basically, anybody that's a QSOFA below a 1 or 0 might not actually be stratified at the right place, because QSOFA is not a very good indicator of sepsis, as we all know. So maybe that patient that was identified as a 4 less urgent should really be reassessed and identified as a 2 in order to see a doctor faster. And of course, patients get discharged, waiting room, or they get admitted to the hospital. So effectively what we do is, this is our proposition, we effectively blend a measure of PSP into existing risk assessments, whether it's NEWS2, whether it's a QSOFA. The idea here is to completely blend into the workflow, and within 7 minutes, by the time you're actually doing your patient introduction, presentation, you do a QSOFA, NEWS2, one or the other, effectively you combine the two, and this is where we've demonstrated a real clinical, proven clinical utility with up to 72 hours, as I mentioned before, predictive value, and effectively it enables a triage nurse to do a rule-in within minutes. What this effectively does is we've identified that a NEWS2 basically will pick up possibly 1 out of 5 septic cases in the waiting room using basic NEWS2 rules. What we can do by adding a measure of PSP is we can actually increase that by 50%. And we will find for those cases that are below 7, we effectively can increase the sensitivity of NEWS2 to up to 33%. That means that the nurse, with that tool in their hands, using existing tools, adding a PSP, can be far more effective at identifying more sepsis cases in the waiting that need a potential bundle. This is how we see this rolling out into an ED, and effectively using different systems, different scoring systems. The idea is to effectively attribute the patients in the right bands to make sure that they get the treatment that they require. Where do we position ourselves? Well, now it's clear. everything on the right side of the screen is a lab process. We don't touch any of this. We are not a lab-based product. We are a point-of-care product. And the idea here is to effectively use the results, the test, at the triage facility in the ED to effectively speed up the bundles and create more order flow to the various tests that will have to take place at any rate. So we're not affecting any of this, but what we're trying to say here is a lot of the testing that happens here that can take from an hour up to 24 hours all the way to 72 hours, all of this can get accelerated and get done faster. But not only do we get it done faster, which is better, earlier is better for the patient, but it actually gets done with patients that you would otherwise have missed. And I think this is the important message here is we are very concerned about those patients that end up waiting in the waiting area of the hospital. They get sent home, they don't get treated. They come back 2 days later, they're worse off, and they end up dying. This is what we're trying to address at this point. Who we are, we're just a simple delivery team, engineers, business people, scientists, biologists. The company's been around for 14 years. It's taken us a decade to basically build this. This may sound like a long time to some of you. That's what it takes to build a new technology company. It came out of the EPFL, École Polytechnique de Lausanne. Some very, very smart people that spent better part of 14 years building this. And we've spent effectively the last 5 years just building up the evidence that we needed for PSP. We realize PSP is unknown to most. There's a big push that needs to be done in terms of physician education, nurses education, et cetera, and not only this, but at some point also involve some of the payers that are part of the procedure, because then they end up having to finance and fund these tests, and as well the patients. So it's a complicated story, but I think it's hopefully a good story that I'm certainly happy to take a few questions and answers if any of you have any. Thank you.